This is the first lecture in neuroanatomy, where we are going to do introduction to neuroanatomy, meninges, and cerebrospinal fluid. Now I'm going to break up this lecture into two parts. The first part is going to be an introduction to neuroanatomy, and the second portion is going to be on meninges and cerebrospinal fluid. Now let's begin by looking at the nervous system. Now the nervous system is divided functionally into two parts. Those are the somatic nervous system, and the second is the autonomic nervous system. The somatic nervous system is responsible for supply of skin and the musculoskeletal components in the body. The autonomic nervous system is responsible for supplying smooth muscle present everywhere in the body and glands. The autonomic is a totally motor system, which is why it supplies smooth muscle and glands. And in the glands, actually, there is a small muscle called the myoepithelial cell. So that's known as the myoepithelial cell, and that's what the autonomic component supplies. The autonomic nervous system is further divided into a sympathetic component and a parasympathetic component. So all of this, the somatic, the autonomic, and the autonomic's sympathetic and parasympathetic divisions, these are all functional components of the nervous system. Now, while the autonomic is a motor system, the somatic is both sensory and motor. And we'll look at this in a little more detail later on. Now, the geographical or the structural division of the nervous system is the central nervous system, which is present in the central axis of the body. So if you look at the central axis of the body, you have the brain and the spinal cord. So that forms the central nervous system. So we have the brain and spinal cord. And we can think of this as the processing center. So this is where impulses are either received or impulses are sent out from here. So this is the processing center. And then from here it is sent to the rest of the body, or it receives from the rest of the body by means of what is known as the peripheral nervous system. And this peripheral nervous system consists of spinal nerves and cranial nerves. So it's the nerves which are present in the peripheral nervous system. So from here, all these nerves will pass out to the body. Now, if you look at this chart carefully, you notice that both the somatic and the autonomic nervous systems have components present within the central nervous system as well as the peripheral nervous system. And quite naturally, they have to have this, because remember, this was the processing center and this was the one that distributed. So this was the distributing center. So for the somatic nervous system, the entire central nervous system is its processing center, and the spinal and cranial nerves are its distribution center. For the autonomic nervous system, since it has two divisions, you have a sympathetic and parasympathetic. The sympathetic does not occupy the entire central nervous system, and neither does the parasympathetic occupy the entire central nervous system. And similarly, the distribution again is not through the entire peripheral nervous system in both divisions. And we'll see this in a little more detail in the next few slides. So let's move on to the next slide here, which shows you the central nervous system and the peripheral nervous system. So you can see here that this part is the brain and the spinal cord. So this is in the central axis of the body, and that forms the central nervous system. And the peripheral nervous system is composed of the cranial nerves, which exit from the brain and move out through the foramina in the skull, and then the spinal nerves exit through the intervertebral foramina present in the vertebral column. Now this gives you a good idea of how the distribution occurs. So let's first look at the somatic nervous system. And I said that it occupies the entire central nervous system. So the entire brain, so if I was to draw the brain up here and the entire spinal cord all the way down, all of this forms the central nervous system component. And then it is distributed by means of 12 pairs of cranial nerves. So 12 pairs of cranial nerves take impulses from the brain or to the brain, and these cranial nerves serve the head and neck region. So they go to the head and neck area. The spinal cord has connections with these spinal nerves, and then there are 31 pairs of spinal nerves, and these spinal nerves go to the rest of the body. So you have these going to the rest of the body. The head and neck is served by the cranial nerves, and the rest of the body is served by spinal nerves. So again, I'm going to repeat this. The somatic nervous system has a central nervous component and a peripheral nervous component. The central nervous component is the entire brain and the spinal cord. That means all of the brain and the spinal cord receives and sends out impulses. The entire CNS is used up as the processing center. It is distributed through the peripheral nervous system by means of cranial nerves and spinal nerves. There are 12 pairs of cranial nerves, and then there are 31 pairs of spinal nerves. And in these 31 pairs of spinal nerves, you have 8 cervical, 12 thoracic, 5 lumbar, 5 sacral, and 1 coccygeal. So that's the somatic part of the nervous system. Let's look at the autonomic part. And we said the autonomic was divided into a sympathetic and a parasympathetic. Now the sympathetic part occupies only the thoracic and the upper 2 lumbar segments of the spinal cord. So you have the thoracic and the upper 2 lumbar segments of the spinal cord, as you can see over here. So its central nervous system component is the thoracic and the lumbar part of the spinal cord. So there is no component in the brain. Its peripheral nervous system is through distribution through all of the spinal nerves. So all of the spinal nerves take impulses and distribute it through to the rest of the body. No cranial nerve is involved in the sympathetic nervous system. It is not involved directly. There's an indirect involvement. So for now, you can just think of it as just the spinal nerves. In the case of the parasympathetic nervous system, the central nervous system component resides in 2 areas. One part is in the brain, and the other part is in the sacral part of the spinal cord. So there's a part in the brain and there's a part in the sacral region of the spinal cord. So you have the brain and the sacral part of the spinal cord. So the brain is in the cranial cavity, so we could call this the craniosacral outflow for the central nervous system. And in the sympathetic, we'll call it the thoracolumbar outflow. Now the peripheral nervous system distribution for the parasympathetic is by means of cranial nerves, and there are 4 cranial nerves which distribute it, and those are the 3rd, the 7th, the 9th, and the 10th. So in the peripheral nervous system you have 4 cranial nerves, the 3rd, 7th, 9th, and 10th, and then you have spinal nerves, and these are the sacral spinal nerves, and these are sacral 2 to 4. So you can see that it has only a few cranial nerves which help in its distribution, and only a few spinal nerves which help in its distribution. So if we were to review this again, we would say that the sympathetic nervous system, its central nervous component, is called the thoracolumbar outflow. Because that's where it originates. And its peripheral nervous system is through all the spinal nerves, all the 31 pairs of spinal nerves. The parasympathetic part, its central nervous system, we can call it the craniosacral outflow. Cranio for brain, sacral for the sacral 2, 3, 4 segments, and its peripheral component is through 4 cranial nerves, which I've already mentioned, and the sacral spinal nerves, and S2 to 4 sacral spinal nerves. So that's the distribution of the parasympathetic and sympathetic. Now let's move further and look at the functional unit of the nervous system, which are where you call them the neurons, and these are the ones which conduct the impulses. And under neurons you have 3 basic types of neurons. The most predominant is the multipolar neuron, which is seen in the brain, in the gray matter of the brain, and in the gray matter of the spinal cord. So this is seen in the gray matter of the brain and the spinal cord. And as you can see, a multipolar neuron, in any neuron for that matter, you have something which is called the cell body or the soma, and then you have processes. The smaller processes are known as dendrites. They're called dendrites. And these dendrites can be from 1 to many. And then you always have just one long process, which is known as the axon. So in a multipolar neuron, you'll have several dendrites and one axon. The second type of neuron that you see is what is known as a bipolar neuron. And a bipolar neuron is seen in all the special sense organs. For example, you will find this in the retina of the eye. You will see it in the internal ear, in the special sense organs of the ear. You will see it in the olfactory epithelium, which is the epithelium of the nose, which is responsible for smelling. You will see this in the taste buds. So these are bipolar neurons seen in special sense organs. And as you can see from this image, it has 2 poles. So there is one dendritic process and one axonal process. So that's why it's called bipolar, 2 poles. And then lastly, we have something which is known as pseudo-unipolar. Unipolar neurons are rarely seen in the human brain. There is only one area where they are seen. So for all practical purposes, we don't see unipolar neurons. Instead, we call it pseudo-unipolar. The reason being, it starts out as having a cell body like every other neuron, and then one process. But that one process soon divides into 2. And one becomes a dendritic process and the other one serves as an axonal process. So that's why this is known as a pseudo-unipolar neuron. And these pseudo-unipolar neurons are seen in the peripheral nervous system, they are seen in what is known as the dorsal root ganglia. These are related to the spinal nerves. And then they are seen in the sensory ganglia of the cranial nerves. So these are seen in the sensory ganglia of cranial nerves. And then the dorsal root ganglia of all spinal nerves. Not all cranial nerves have sensory ganglia, but those which do, they are seen in those sensory ganglia. Now let's look at the structure of a neuron in a little more detail. And here we can see this is showing you a multipolar neuron taken from the gray matter of the spinal cord. So you can see that this is the cell body, and if you look at the cell body a little more clearly, you have the cytoplasm, and then inside the cytoplasm you have tiny filaments and tubules present, which you can see here as thin lines. These are, and you can see them up here too, the neurofilaments and microtubules. And then in the cell body you'll see a nucleus, a prominent nucleolus. So the nucleolus is very, very prominent in a neuron. That's the nucleolus. And then you see dark staining structures which are known as Nissel's granules or Nissel's bodies, and these represent rough endoplasmic reticulum. So this is a site for protein synthesis. Rough endoplasmic reticulum synthesizes proteins, and so these Nissel's granules which are present here, these represent rough endoplasmic reticulum. Then you can see that these are the dendritic processes. And then of course you have one axonal process present here, and in this case the axonal process has a myelin sheath, which we'll learn a little bit more about myelin. Now also seen in this image is when one neuron makes contact with another neuron, we call it a synapse. So let's say you have one neuron, one multipolar neuron, and that has an axonal process. It makes contact with another neuron, and this region here is known as a synapse. There is a cleft present here, and most synapses in the brain are what are known as chemical synapses, because this neuron which is present before the synapse is known as a presynaptic neuron, and this neuron which is present after the synapse is known as a postsynaptic neuron. And these presynaptic neurons will release a substance into this synaptic cleft. That substance is called a neurotransmitter. That neurotransmitter will come and attach to receptors present on these processes, and then that will depolarize the membrane in most cases, and the impulse conduction will continue and then go on to the next one. Now this is a typical picture that you see where you have an axon synapsing on the dendrites of the postsynaptic neuron, and such a synapse is known as an axodendritic synapse. And most axodendritic synapses are excitatory synapses. Sometimes you might have a synapse in which the axon, instead of synapsing on the dendrite, might actually come and synapse on the cell body, on the soma. So such a synapse will be known as an axosomatic synapse. And then rarely you have synapses which are known as dendritic, where a dendrite will synapse with another dendrite, or you have a synapse where an axon will synapse with another axon, and these are known as axonic synapses. Now, as I mentioned earlier, you had neurotransmitters which were released at these synapses. There are different types of neurotransmitters. Some common ones have been given to you in the notes. And one important thing to remember is that in the central nervous system, you must know some of these names. In the central nervous system, the commonest excitatory neurotransmitter is one which is known as glutamate. And the commonest inhibitory neurotransmitter is known as GABA, which is gamma aminobutyric acid. Gamma aminobutyric acid. So this is excitatory and this is inhibitory. In the peripheral nervous system, you don't really have any inhibitory neurotransmitters, but the neurotransmitters for motor neurons to skeletal muscle is acetylcholine, and this is how it's usually written. And the sensory neurons, they use the neurotransmitter glutamate. So sensory neurons use glutamate and motor neurons use acetylcholine in the peripheral nervous system. Now let's look at some terms in neuroanatomy which are important, and we'll be using these terms frequently, and hence you must be familiar with these terms. First, let's look at terms related to collection of neurons, which means when I say neurons, I mean nerve cell bodies. Now, a collection of the nerve cell bodies in the central nervous system is known as cortex, or also known as gray matter. It's called the cortex in the brain, and especially in the cerebral hemisphere, where you can see that it's lying along the periphery forming a thin shell. So this area is known as cortex. You could also call it gray matter. In the spinal cord, this collection of neurons is not present in the periphery. Instead, it comes into the center. So it's present here. And here we just call it gray matter. We don't give it the name cortex. So when you use the word cortex, you're still talking about collection of nerve cell bodies. You could call it gray matter, but the word cortex is used typically only for the brain. If you were to refer to a collection of gray matter in the spinal cord, you wouldn't call it cortex, you would only call it gray matter. Now, gray matter is also seen in the brain as little islands, and those areas are known as nuclei. So those areas are called nuclei. And the reason is that initially, while the brain was developing, initially when the brain was developing, so let's say in the primitive brain, when the brain was developing, the gray matter was actually present right in the center. And then as evolution occurred, you wanted to increase the number of connections made so that the person or the animal could perform more functions. So first of all, the brain increased in size, and then the brain got convoluted, which means, it sort of had all these little ridges and small furrows present. So this gray matter then migrated to the periphery, and it was now present along the periphery as the cortex. So you can see that it increased a great deal, because as it kind of went in this way, the amount of gray matter increased. Some of these islands remain, some of this gray matter remains in the core of the brain, and instead of being represented as one big mass as you see here, it was represented as small little islands of neurons, and these are known as nuclei. And many of these nuclei have names within the brain. For example, you'll have a nucleus which is called the red nucleus, and then you have something known as caudate nucleus. You have another nucleus which is given the name of thalamus. So you have many of these which have been given names, and that's what we are looking at over here. So if you look at this image here, if you notice, caudate nucleus has been identified. The putamen is again a nucleus. There is another area of gray matter called the claustrum. They're given different names, and as we go along, we'll be doing that. So we have cortex, we had gray matter, and we have nuclei. All of these are collections of nerve cell bodies. Another term for collection of nerve cell bodies which you will see in the brain is substantia, in the brain and spinal cord. So substantia is another term which is used for collection of nerve cell bodies. And here again you can see lentiform nucleus, so another name for nucleus. In the peripheral nervous system, a collection of nerve cell bodies is given the name of ganglion. And if you remember, I referred to the ganglion earlier. I called it a dorsal root ganglion. Dorsal, another word for dorsal is also posterior root ganglion. So you have this posterior root ganglion in relation to the spinal cord. And in relation to cranial nerves, you'll have what are known as sensory ganglia of the cranial nerves. So ganglia is another term, or ganglion is another term which tells you that this is a collection of nerve cell bodies. Now, when we have collection of processes, and usually those processes are axonal processes, we use a different term. We call it white matter. And you can see in the brain, this white matter is more central. So this is white matter. Whereas in the spinal cord, the white matter is more peripheral. So you have white matter, which is a collection of axons. The collection of axons can also be known as tracts. And usually when you refer to a tract, these are fibers which have a similar function and a similar direction. That means they arise from one particular area and they all terminate in another area, in a similar area. So tracts is another term for collection of axons. Lemniscus is another term which you will see, which is again a term used to describe a collection of axons. And you'll be seeing all of these. So white matter, tract, lemniscus, these are all terms used for collections of axons. Let's move on to the next slide. And here we are going to talk about, once we've done the nerve and we've talked about, the various parts of a neuron, talked about axons, let's look at some functional aspects of it. Now we said inside the soma, remember we said we had the Nissel's granules, we had rough endoplasmic, which was the rough endoplasmic reticulum, we had all those microfilaments and microtubules. Now much of the processing occurs within the soma, and then it has to be transported towards the synapse. So this transport which occurs from the soma or from the cell body down the axon and usually towards the synapse, this type of transport is known as anterograde transport. And this anterograde transport is used basically for transporting neurotransmitters which are produced here and they travel all the way down, for transporting proteins, mitochondria needed towards this terminal end of this axon. So this is used for transport of all this material. The second type of transport is when the transport occurs in the opposite direction. So it occurs this way. And that type of a transport is known as retrograde transport. And retrograde transport is usually used for waste disposal. So anything present in the synaptic cleft is kind of taken in by endocytosis. And then it's taken to the cell body where it is disposed of. And it's also very important because you have something which is known as neurotrophins or nerve growth factor. So this is known as nerve growth factor. and this is taken in at the synapse, and this is very, very important because it is needed for the development of sensory and autonomic nerve fibers. And what really happens is in this retrograde transport, and if you look here at this part, at this synaptic contact, which is here, this is an axon and this is a dendrite, and this area here is the synaptic cleft. These axons, they constantly nibble. It's almost like they're nibbling the plasma membrane of these target neurons. So this one here is the target neuron. And they nibble at this target neuron, and by endocytic vesicular uptake, you can see these are little endocytic vesicles, they are taken back in the opposite direction towards the cell body, and what they are really taking up are what is known as marker molecules. A lot of this is done during development, and this uptake of these marker molecules is important for cell recognition during development, so that, nerve cells know with which cells they need to establish and maintain their contact. So that is retrograde transport. Now, why do we need to know this anterograde and retrograde transport is because we can explain a lot of diseases based on this. So the clinical applications are, most of it involves retrograde transport, and the first one is in the case of tetanus, the disease tetanus, which is through the tetanus bacteria. Now that liberates a toxin. So when one injures themselves, and let's say the tetanus toxin is, finds its way through the skin, and then through the skin the toxin binds to the plasma membrane of nerve endings and is taken up by endocytosis, and it travels, so from the skin area it travels in a retrograde fashion towards the spinal cord. So here this is the spinal cord and this is the peripheral nerve. So it travels, as you can see, in a retrograde fashion to the spinal cord, and then from the spinal cord again it is taken upstream. It's taken by special cells, and these cells are known as Renshaw's cells. And these cells are known as Renshaw's cells. And the function of these Renshaw cells is normally these Renshaw cells which are present in the spinal cord. So let's say that this is a Renshaw cell, and this is present within the spinal cord. This Renshaw cell synapses on other cells which exit the spinal cord. So this tetanus toxin goes in, and it's particularly taken up by these Renshaw cells. And these Renshaw cells normally have what is known as a braking action, a braking or an inhibitory action on these motor neurons. So they kind of keep a check on these motor neurons, and they prevent these motor neurons from firing incessantly. The way they do that is these Renshaw cells liberate a neurotransmitter which is known as glycine. Now when the tetanus toxin is taken up, it goes through retrograde transport and it goes to these Renshaw cells. What it does is it knocks these Renshaw cells off, and hence these Renshaw cells can't produce glycine. These motor neurons no longer have this braking or inhibitory action. So what happens is that they go out of control, and that's why in tetanus you find that the muscles tend to twitch continuously, and most of this takes place in the muscles of the face and the spine, and that's why the patient exhibits prolonged agonizing spasms, and most patients just die of exhaustion. So that is how the tetanus toxin acts through retrograde transport. Then we have viruses and other toxic materials. Most viruses present in the nasopharynx travel in the nasopharynx. You have a lot of viruses which are present in the nasopharynx. They travel to the central nervous system, and certain toxic materials like lead and aluminum, they also can travel in a reverse direction or retrograde fashion and go and affect the nervous system. Other viruses like rabies, polio, the poliomyelitis, herpes, again use this retrograde fashion of travel. And in fact, in rabies, it's seen that the incubation period is proportional to in rabies, the incubation period is proportional to the length of the nerve, of the spinal nerve, or cranial nerve for that matter. So the longer the nerve is, the longer the incubation period is. So if somebody was bitten by a rabid dog on the leg, and there was someone who's bitten by a rabid dog, let's say on the arm, the person with who's bitten on the arm will show signs or symptoms of rabies much sooner, because the nerve from the leg takes much longer. It's a longer nerve traveling to the spinal cord. And again, as I said, herpes and polio also are believed to spread through this retrograde type of axonal transport. Now, the anterograde type of transport is involved in what is known as peripheral neuropathy. And in peripheral neuropathy, what usually happens is that there's a defective anterograde transport. So the distal parts of long peripheral nerves, they undergo atrophy. So usually the person exhibits what is known as a glove and stocking, because it's the distal part of the peripheral nerve. Glove and stocking type of affectation. So the problem occurs in the hands and in the feet. And this is because of a defective anterograde type of transport. Now, while we are talking about neuronal transport, and we mentioned synapses, I'd also like to mention at this point that how general anesthetics and local anesthetics act. General anesthetics, they block synaptic transmission. So synaptic transmission does not occur, and hence that's why you can see, the person has analgesia and anesthesia. They kind of get drowsy. Local anesthetics, what they do is they act on the axolemma, or the cell membrane of the axon, and they interfere with the permeability of the axolemma to sodium and potassium ions. So they interfere with this. So you remember depolarization, you need sodium to go in and potassium to come out. So they interfere in the permeability of the axolemma to sodium and potassium and other ions. And what happens is that the sensitivity of nerve fibers to local anesthetics is proportional to the size. Smaller fibers are more sensitive to local anesthetics, and pain fibers are usually much smaller than any other fiber, which is why when you apply local anesthetic to any area, the pain fibers are the first to go, and that's why you don't feel pain at that point, or that area becomes numb. Now let's look at the supporting cells, which are very important. These are the supporting cells. They do not take part in nerve transmission, but they support the neurons. And actually these are known as nerve glue, and they outnumber the neurons. In fact, they are 5 to 1. The supporting cells, for every one neuron, there are 5 supporting cells. So they are 5 to 1. Now the different types of supporting cells that we have are astrocytes. These are, as you see here, these are astrocytes, and the astrocytes are important because these are nutritive in function. They contain glycogen granules inside them, and glycogen is an immediate source of nutrition for the neurons. Plus, the astrocytes also have these vascular processes. If you look up here, you can notice these little vascular processes which are sort of attached to this blood vessel, and the other end of these processes are attached to a neuron. So they take the nutrition from the blood vessel and transfer it to the neuron. So these vascular processes again help in nutrition. Astrocytes also recycle. They also recycle neurotransmitters. So they take the neurotransmitters from here and take it to the blood vessel. So they recycle neurotransmitters. And during the healing process in the central nervous system, they form dense scar tissue. So they form dense scar tissue during the healing process. They are able to proliferate quite rapidly. So they form scar tissue. And in fact, local proliferation of these astrocytes is often responsible for the formation of brain tumors. And, one of the common types of brain tumors is what is known as an astrocytoma. So these are some functions. They have glycogen molecules which act as an immediate source of energy. Their processes invest capillaries, and on the other hand, they have connections with the neurons. So that's how they transfer nutrition to the neurons. They recycle neurotransmitters. They're involved in healing in the central nervous system, and they form scar tissue, which is really not a good thing, but that's the way they act. They proliferate quite rapidly, and tumors are formed when they have uncontrolled proliferation, and a tumor is known as an astrocytoma. And it is thought they also help to insulate neurons. So that's another function of astrocytes. The second type of supporting cell is what is called in the central nervous system is known as an oligodendrocyte. And an oligodendrocyte is responsible for myelination in the central nervous system. So it lays down myelin in the central nervous system. And a single oligodendrocyte can lay down myelin on about 3 dozen or more axons. So if you look here, you can see this is one oligodendrocyte, and notice it's myelinating this axon, it's gone on to this axon, so it can myelinate about 3 dozen or 60 axons in the central nervous system. And the purpose of myelination is it increases the speed of impulse conduction, because when myelination occurs, it leaves small areas. Over here, you can see little areas which are left bare of myelin, and these areas are known as nodes of Ranvier. So if we have an axon, you will see that the myelin is present here, then it leaves a little gap, myelin is present here, and again here. So these little nodes which are present, these are known as the nodes of Ranvier. And myelination increases the speed of impulse conduction because depolarization jumps from one node to the other. So it jumps from one node to the other. Now, when myelination occurs, the potassium channels are depleted from this area underlying the myelin. The potassium channels are depleted, and hence in some demyelinating diseases like multiple sclerosis, impulse conduction fails because this area is gone. So the impulse cannot jump from this area to this area because the insulation is no longer present, and the potassium channel was not developed to begin with. So that's what happens in demyelinating diseases. The next type of supporting cell we have are known as microglia, and these are the phagocytes of the central nervous system. These are motile, so these are the motile phagocytes of the central nervous system. Then the other supporting cell present in the central nervous system are the ependymal cells, and these ependymal cells which you see up here, these are the ones which line cavities in the brain known as ventricles. So they line cavities, and with the help of blood vessels associated with them, they help to produce cerebrospinal fluid. So those are the ependymal cells. Now, in the peripheral nervous system, so all of these cells which we saw here were present in the central nervous system. In the peripheral nervous system, we have a cell present which is known as the Schwann cell. And here it is. So here is the Schwann cell which is present, and the Schwann cell is the counterpart of the oligodendrocyte. So the function of the Schwann cell is to lay down myelin, but this is laid down in the peripheral nervous system. So that's one difference. And the second is that a Schwann cell does not myelinate many axons. It myelinates only one axon, unlike the oligodendrocyte, which you can see myelinates several axons at one time. Each Schwann cell myelinates just one axon. So one axon may have many Schwann cells. Schwann cells do not myelinate many axons. Now, the reason you see multiple sclerosis over here is because I talked earlier about demyelinating diseases, and multiple sclerosis is one type of demyelinating disease where the myelin is actually broken down. So it's a type of demyelinating disease which occurs in the central nervous system. And here what you have is you have plaques of demyelinations or areas of demyelination. So one area will break down, the myelin will break down, and then the axon is not able to conduct impulses because those potassium channels are normally deleted from that axolemma. Now, this multiple sclerosis is not anatomically selective. So in other words, it can affect both motor neurons and sensory neurons within the central nervous system. So patient will present with either motor deficit or with sensory symptoms. In the peripheral nervous system, one type of demyelinating disease is known as the Guillain-Barré syndrome. And here again you have axonal demyelination, and the patient will present with severe sensory or motor disturbances depending on which nerve fiber is affected. We'll talk about these demyelinating diseases in a little more detail as we do the different types of diseases. Now here let's talk about degeneration and regeneration within the nervous system. And first we are going to look at degeneration. Now there's one type of degeneration which is seen, and this is known as Wallerian degeneration. And Wallerian degeneration occurs in the peripheral nervous system mainly. And what happens is that when the nerve fiber or the axon is cut, then let's say up here, when you look up here, so here the nerve fiber or the axon has been crushed. So you get a proximal stump, and then down here will be this part here is the distal stump. So what happens in this degeneration is that the axon and the myelin, they begin to disintegrate, and the axon, as you can see, begins to disintegrate and forms small little fragments. These fragments are known as ellipsoids. So the Schwann cell also begins to disintegrate, and because of this disintegration, the macrophages which are present in blood vessels surrounding them, these macrophages are monocytes which have got converted to macrophages. They come in and they remove the cell debris. So you can see up here the macrophages have come in and they've started removing all the cell debris. So all the cell debris is cleared, and so then now we have a tube which is formed by the endoneurium. So this endoneurial tube is clear, and all the cell debris have been removed at this point. Now these macrophages, which were originally monocytes and which came in to remove the cell debris, they are really important in the peripheral nervous system, because later on, as you'll see in regeneration, these macrophages, they bring in factors which are known as trophic factors, which are feeding factors, and they also bring in tropic factors, which are guidance factors. So they help to nourish the damaged nerve, and they also help to guide the nerve in its regeneration. So not only the macrophages function to remove cell debris, but they also bring in these trophic and tropic factors. Trophic are nutritive factors, tropic are guidance factors, and so they help in regeneration. And they also help to stimulate these Schwann cells, and again we'll see that in the regeneration. They stimulate these Schwann cells and help them to divide. Now that's what happens to the axon. Now to the cell body, what happens? It undergoes a process which is known as chromatolysis. And chromatolysis basically means that the cell becomes pale, and the reason is that once the axon is damaged, then the nerve cell body swells up, and the Nissls granules, they get distributed throughout. As you can see here, these Nissls granules get distributed throughout the cell, and they go to the periphery, and as does the nucleus. And the nucleus also becomes peripheral, and you no longer see the Nissls granules, and that's why it looks, this Nissls granules the cell looks pale, or that chromatolysis really means loss of color. And in most parent motor neurons, which become isolated from synaptic contacts in the gray matter, they become isolated from synaptic contacts in the gray matter, and that's because of these neuroglial cells in the central nervous system which go and form scar tissue in between. Now in the central nervous system, degeneration which occurs is the macrophages there are the microglial cells, but they remove debris much slower, and in fact the debris can be identified 6 months after injury in the central nervous system, whereas in the peripheral nervous system it's virtually cleared by 6 days. In the central nervous system, chromatolysis is usually unusual. Instead, the cell just dies. It undergoes necrosis, and those which do sort of survive, they appear wasted, and they usually have no contact with other nerves because of this glial tissue, which, if you remember, I told you astrocytes form scar tissue. They help in healing. So when a nerve is damaged, the astrocytes go in and they fill in the gap, and so then the neuron is prevented from making contact with the cells that it had contact with. Let's look at regeneration. Now regeneration occurs quite it occurs in the peripheral nervous system. It occurs quite freely, whereas in the central nervous system, regeneration does not occur, or rather it's unsuccessful. Now in the peripheral nervous system, what happens is that if you look here, this is the proximal stump, and regeneration is possible only if the proximal stump is able to make contact with Schwann cells. And if it doesn't make contact, then what happens is they just grow erratically and form what is known as a neuroma. And you'll see how they make contact with the Schwann cells. So usually in a clean cut wound, the axon, which is up here, it forms little sprouts, and these sprouts begin to be seen in a few hours. In a crush injury, it might take a little longer, maybe delays or a week at the most. And then the Schwann cells also begin to undergo mitosis. So you can see here that the Schwann cells undergo mitosis, and these Schwann cells make contact with these these little sprouts present here. In fact, these sprouts are known as growth cones. So after making contact with these growth cones, they actually pull them and they direct them in a downward fashion. So you can see how they're kind of directing it. If you remember, I said you had those trophic and tropic factors. So this is that guidance factor, and it helps to pull these growth cones down, and as they keep going down, Schwann cells lower down, again undergo mitosis, and they help to pull this down, and this thereby grows within and makes contact with the distal stump. At the same time, you can see the Schwann cells now are beginning to lay down myelin, and then the nerve cell ends up, making contact with the distal stump and becoming fully myelinated. Now, all of this will occur properly only if this main thing, which is this endoneurial tube, is intact, which is why in a crush injury, and if we go back to the previous slide, you can see this is like a crush injury up here. So you can see here, this is a crush injury. A crush injury is much better than a clear-cut, severed neuron, because when a neuron is completely severed, so imagine if it's severed up here, so what will happen is that there is a discontinuity between the endoneurial tubes, and then regeneration is not possible, or rather you'll have to manually do that, and that is by suturing the endoneurial tubes. So when there is a clean cut wound, usually what one does is you wait for about 3 weeks so that the connective tissue sheets would have thickened and they'll hold your sutures well. So that's how regeneration occurs in the peripheral nervous system. In the central nervous system, the one regeneration does not occur successfully. One, because the clearance of debris is very, very slow, and usually the cell dies. And at the same time, you have the glial scar tissue which comes in between. So, if you have scar tissue which has come in, it prevents the nerves from making contact, the axons from making contact. And at the same time, the oligodendrocytes, they produce breakdown products, and that inhibits growth. So all of this prevents regeneration in the central nervous system. At the same time, the central nervous system also lacks those trophic factors, which is why one uses the fetal tissue to promote regeneration in the central nervous system. Because fetal tissue has those trophic factors or nutritive factors, and if you were to implant fetal tissue into the central nervous system, they have found success with regeneration. And that's why research is being done for Parkinson's disease and Alzheimer's and certain other conditions where fetal tissue is implanted into the central nervous system to aid regeneration. So that completes regeneration. So just to recap, regeneration is possible in the peripheral nervous system, and the possibility is only when the proximal stump can make contact with Schwann cells and thereby be guided so that it can go and lead to the distal stump. If it fails to make contact with Schwann cells, then you end up with a tumor or a neuroma formation. Again, crush injuries are better because the endoneurial tubes are preserved in crush injuries, whereas if it is a clean cut wound, then you'll have to suture it. In the central nervous system, because of lack of those, there is a lack of tropic factors, so there's a lack of these tropic factors. The oligodendrocytes produce breakdown products which inhibit growth, and because the debris is removed slowly, regeneration is not possible, which is why you use fetal tissue to help promote regeneration in the central nervous system.